Hello friends, welcome you in Research Needs. In this video, we will see how to use novelty suppressed feeding test for depression study. So for these experiments, we need some tools and equipments. For this study, we need a big card box. It should be 100 centimeter long, 100 centimeter wide, and almost 50 to 40 centimeter in height. And it should be painted brown so all the sides, and a webcam should be attached to the ceiling and connected to the recording device, and especially software which known as a motion tracking software or device. And next thing you have to consider, you have to check how much the light is present in different parts of the box, and it should be 1000 lux around at the center of the box where we are uh, putting the feed platform, and at the side of the boxes it should be around 100 lux illumination, and the floor of this box should be wooden material and that we can easily clean after each animal test, so that you have to maintain. In the middle of the box we should put the little bit the tasty food while we are performing this experiment for the animals that they are finding during the experiments. So next, if you need this kind of video for your experiment, you have to subscribe Anyways software, because in that software you easily get the motion tracking which easily give this kind of results. So before you start experiment, animals should be acclimatized for 5 days for 10 minutes every time in this box for each animals, and this experiment generally performed for 10 minutes. After completion, animals should be provided food because means while you performing this experiment, before that you have to keep animal for 24 hours fasting, especially while you using rats. In case of mice it should be 6 to 12 hours. In this experiment, motion tracking software should be able to record how long it took for the rats to go to the food for the first time. So it means first food touch latency time you have to record. Second, how much distance is being traveled by the animals, that should be measured. And third, quantity of feed eaten by the each animals. Means you have to record the feed latency. Means how much quantity feed each animal has eaten during the experiment. So these parameters you have to record while you performing this experiment. So this is all about novelty feed suppression test. So now we are going to see when and where we are using this experiment, means this type of test. So generally we are using this type of test in depression study, and other parameters are other tests are force swim test and tail suspension test. That kind of parameters we are taking while we are doing depression study. And before performing this experiment, generally we are performing this experiment on day first while we are starting the experiment. Means we are starting the depression study. So first time we are taking the baseline parameters. In that we have to perform this experiment before starting the experiment, means on day 0, and finally on day 56 after 2 months later we are performing this experiment after complete induction of the depression. Means In between, we have to give the stressors to the animals. Now we will see what are these stressors. Some of them I'm going to explain you here. So while you are performing this experiment, you have to perform this around 8 weeks to induce depression. And each weeks you have to divide each in every week you have to divide. So you have to give 7 stressors each day. Suppose first day you have taken empty drinking bottle. You have to keep means your in animal cage. You can remove the water and you have to keep the empty bottles for the animals. So this way animal are not getting water for whole day. So this kind of stress you have to give. So this is first stress. And in second kind of stressor, very second day you have to do limited the food, providing limited food means you have given if animal consuming for 3 animals consuming around 100 grams, so that day you are providing only 20 grams. So whole day you have to keep like that, only 20 grams feed you have to keep. So this is limited feed feeding stressors. And next day you have deprived the whole animals with the uh, food materials. This is third day you are performing this kind of stressors. And fourth day, for 4 hours around you have to give 100 decibel noise to all animals to whom you are kept in the experiment. So to whom you are reducing the depressions. And next another fifth day you are providing complete illumination for whole day. Means you have started light for 24 hours. Like that you have to maintain. So fifth day you are performing this kind of activities. And on sixth day for 12 hours you have added 100 or 200 ml of water in their cage. So all the bedding material will get wet. So this type of way they have to means spend their whole day. So this is the sixth stressors you are providing to animals. On seventh day, suppose you are uh, adding all animals in uh, 4 degree temperature water. They at that time they have to means in that water, yeah, they have to swim forcefully and for around 10 to 15 minutes or 1 hour. So this kind of stressor you have to give. So you have to see if the weather is supporting, then you have to perform this kind of experiment. Suppose while you are putting the animals in the cold water, that time you have to check the weather is supporting to animals health or not, because sometimes animal get die. So these kind of stressor you have to provide animals to produce depression. So next you have to perform SPT, sucrose preference test. In that you have to see that SPT will give you idea if animals are induced with depression or not. If you control animals and normal control animals and disease control animals are not showing difference, significant difference, then you have to perform this depression study. Means you have to induce, you have to give more stress to your animals. So these are the parameters by which you can perform the novelty feed suppression test. So how was the video? Give me the uh, your feedback by comment. And if you want to know more about this depression study, you can write comment. Thank you for watching this study.